some of the projects I'm presenting here.
Wow.
Okay. The work I'm going to be presenting here spans quite a number of years. I must also say that no part of the work has so far been published for very good reasons. And I believe as we go on, you understand why that is the case. Although that project is going to be later on in the presentation. At the beginning, we're just going to talk generally. We consider the problem at hand, and we'll also try to do justice to explaining why I have chosen some of the terms, some of the words that I've used in describing the title. Okay, I am Olujide Olubiyi. I'm a pharmacist by profession, and then my research area, training and research is mostly in the area of drug discovery generally, and especially in computational drug design, the use of computers in the discovery of new lead molecules. And I'm at the Department of Pharmaceutical and Medicinal Chemistry, College of Pharmacy at Babalola University. So the topic today is malaria, the Mephistophelean Behemoth, rethinking drug discovery strategies in Africa. There's nobody that's going to be in the class today that is going to be a stranger to malaria. Okay. I also want to not guess, I just want to project that everybody in the class today has had malaria at one time or the other. In fact, it has become we have become so used to the concept of malaria and to the experience of it in this part of the world that it has become a second nature. A lot of times when we talk about malaria, it rarely evokes the sense of dread that COVID-19 evoked when it was around. And the funny thing is that malaria is a lot much more dangerous than COVID-19. The question is that how come it never really evoked that kind of dread in us? Is it because it affects Africans generally? I'm not going to be the one to answer that question. So malaria is not something that we just got to know a few years ago. It had actually been in existence for thousands of years. The brief history that I'm trying to put forward in the slide here is just trying to cut that history to the beginning at the 18th century, but assure us that it has been in existence for more than 3,000 years at least, even though it might not have been called malaria. Okay, it was derived from the 18th century Italian swamp miasma theory. Ancient civilizations attributed the shivering fever, what we call chills, to noxious marsh vapors. That was at the beginning. And then it was much later that Ronald Ross was able to identify the Anopheles vector, the mosquito, in 1897. Now, why did I choose the word Mephistophelian and Behemoth? The Phocian demon of deception, that is what they call Mephistophelian. In fact, in German legend, the name's supposed to be synonymous with the devil. Okay. There was a story of a very bored writer. He was an author, but he's bored. He had no ideas and all that. So he came in contact with Mephistopheles. Mephistopheles promised to give him anything that he would want. And he agreed. The man agreed. So Mephistopheles gave him the ability to write good books, to be rich, to be wealthy, to have everything in this world that he wanted, but in exchange for his soul. Now, why am I choosing this word to describe malaria? The reason I've chosen this word is that a lot of times you will look at the kind of malaria research we have been doing, not just in Nigeria, in more parts of Africa, and even in the world. We usually focus on the discovery of new molecules, just like we do for most other drugs. At the moment we're able to get a new molecule, one or two of them, we are going to get something good, and then everybody's happy for some time. So every single drug therapy that has been coming in the area of malaria has always been a temporary cure. I remember when artemisinin came out and it was like the magic drug. But now, anybody that knows anything about malaria knows that the party of ACT is almost over. Okay, because we have development of resistance in some parts of the world. It has even got to Africa against these molecules that have been so beautiful. And why behemoth? Behemoth talks about a monster that is very difficult, very big, very monstrous, and difficult to overcome. And if you look at it too, you will see that we have in malaria a parasite that knows exactly how to navigate its way. I mean, genetically, it's able to transform, to adapt to every single new challenge that we are throwing at it. So that's the reason why I call it Mephistophelean. And Behemoth is because it's so big, it's so devastating, especially in Africa. A lot of times you have people in your family who has malaria, they just take some drugs and they're okay. And you believe that malaria is not so bad. But by the time you're looking at the statistics, you we have an idea that it's worse than we think. For instance, when we're talking about the behemoth, the parasite Plasmodium falciparum, it has over 5,300 genes, and it has a lot of ability to vary any part of its genes to be able to navigate any drug challenge we throw at it. And of course, we have had a lot of public health, let me say, interventions. We have had the rollback malaria program at some point, where the government of Nigeria was subsidizing malaria treatment, especially to children below the age of 5 and to pregnant women. They were getting them for free. At another point, they started charging a token, maybe 30 naira and something like that. And the funny thing is that even though that particular method, that particular approach was massively useful. But at the same time, I personally believe that it also spurred the development of resistance to the molecules that we have. And that's because generally when people get drugs for free, they don't respect it. If you're supposed to use artemisinin, let's artemether lumefantrine, you're meant to use it for 3 days. Most people they use it for 1 or 2 days. By the time they get a little bit relief in their symptoms, they dump it. Why won't they dump it? Because it was free. And by the time you use anti-malaria drugs against this parasite we're talking about, and you do not complete it, you are actually begging the parasites to develop resistance. So a lot of people have succumbed to malaria. We have Alexander the Great, although that's a little bit conjectural. And that's because some people believe the acute fever that killed him might not have been malaria. Some people thought it was poison, but in some cultures, they believe malaria was involved. We have Dante Alighieri, that's in 1321 AD. We have a pope, the Pope who lived the shortest time in the history of Catholic Church. He lived only 12 days because he died of malaria soon after his election. Lord Byron, too, a number of them. The thing is that the list is endless because the fatalities due to malaria fever is easily in the millions. But these are the very notable people that we know. Now, let's quickly talk about milestones in malaria scientific discovery. We had Afonso Levaran, who discovered the parasite in red blood cell of a soldier. That was in North Africa, that's in Nigeria, and he got the Nobel Prize for that. And by the time the people could pinpoint that there was a parasite, there was a parasitic cause for malaria, it became a lot easier to tackle the problem because before that time, the people were attributing it to a lot of things, spiritism, I mean, religious reasons, let me say, but by the time the scientist community started asking the questions, started giving the, how do I say now, uh, objective reasons why malaria was developing. For instance, uh, mosquito parasite, mosquito vector is involved, then there's plasmodium parasite involved. So this was discovered by Afonso, got Nobel Prize for that. We have other people who also work on that. Then the very interesting story that I always find very interesting personally is the story of Tuyuyu, who in 1972 who was able to discover the plant Artemisia annua and extract artemisinin from it, King Gaozu. And then this fetched her a Nobel Prize almost 30 years after. That was just about in 2015. And thank God for the discovery of this woman. She's a Chinese woman. Millions and millions of people have been saved from succumbing to the fate of malaria. So if we were to examine the epidemiological data, the World Malaria Report data in sub-Saharan Africa, when we say sub-Saharan Africa, we're not talking about the whole of Africa. We're just talking about the southern part of Africa. I don't mean South Africa, but just the southern part, just a little bit below the equator downwards. You know, that's what we are referring to. So in sub-Saharan Africa, we have a disproportionate brunt that we are bearing of the malaria problem. For instance, the global malaria cases were about 249 million people affected, and 94 million of global cases is concentrated in sub-Saharan Africa. Just imagine that for a minute. If you were to calculate 94% of 249 million, you are going to get 233 million. That is about the size of Nigeria who are affected by malaria. And 95% of global death, everybody that dies from malaria, about a little bit above 600,000, annually, they die in Africa. Just imagine that that's more than 500,000 people die in Africa because of malaria. And you should also think that it is in spite of all the therapeutic options that we have, in spite of all the existing drugs and all the existing public health campaigns and programs. You understand? We still have more than 500,000 people dying in Africa alone every single year. And the most vulnerable people, they are the children under 5. Children under 5 account for about 80% of all malaria deaths in WHO African region. The statistics is that every single minute, I think we have gone into this talk for about 5 minutes now. Every single minute, a child dies. So it means about 5 children would have died because of malaria just in the space of 5 minutes that we must have spent together. And if you consider the socio-economical drain, we can see the figure here that about $12 billion is directed, is the economic loss annually from Africa. The question is, how rich is Africa that we are losing this amount of money because of a single disease? Okay. Now, let's come to Nigeria. We're talking about Africa generally. About 27%, and Nigeria holds about 27% global share, not just Africa now. And then we have about 31% global death is recorded in Africa. We have a little bit about 600,000 people that die from malaria every year globally, but about 31% of this number, I think we should be a little bit above 200,000 people occurs in Nigeria. After this, we have the Democratic Republic of Congo, about 12%, and then we have Uganda, about 5%. So there is urgency for Nigerian researchers. This is not the question of just the government now. This is a task for every researcher in life sciences, not in pharmacy, not in medicine, not in microbiology. Every single researcher in life sciences, now we have a call that we have a disease that we must together start looking at and see how we can bring it to an end. So with over 67 million annual cases and 190 thereabout deaths locally in Nigeria, we require indigenous innovative approaches to be able to dismantle this parasite's stronghold on Africa and on the world generally. So if you want to let's just consider a little bit of the Plasmodium biology and the species that affect humans generally. There are 5 of them, 5 Plasmodium species. We have the falciparum, which is the most virulent, and it causes microvasculature, sequestration, cerebral malaria, and is responsible for over 90% of African mortality that is due to malaria. Sometimes you wonder, why is it that this is what we have in Nigeria, the worst of all the Plasmodium species? Then we have the vivax, which forms dormant liver hypnozoids and predominant in Latin America, Asia, and expanding into Africa, especially in the Horn of Africa. We have Plasmodium malariae. We also have Plasmodium ovale and knowlesis. Okay. The life cycle is a little bit it's not a little bit, it's actually a lot complex. Okay, we have the mosquito stage where the female Anopheles injects the sporozoites during blood meal, and then you have this sexual reproduction, sporogony occurs inside the vector gut. The interesting thing is that it's only the female Anopheles that actually feeds on blood meal. It's not because it needs it. Mosquito generally feeds on sugary things, nectar, that's the food. But the female Anopheles mosquito needs the protein and the constant that comes from human blood meal or blood meal generally to be able to feed its young ones, to be able to reproduce. And that's why it always comes for the human blood. So you also have the liver stage and you have the blood stage. The liver stage, the sporozoids, they invade the liver cells, the hepatocytes, they multiply into tens of thousands of merozoites. And then in the blood stage, you have this merozoid bursts into the bloodstream. It affects red blood cells, and you have several phases in that too. And this is where you begin to have the symptoms, the clinical symptoms of malaria. I'm sure if we were to share our personal experiences, all of us, we don't even have to read any book or study anything to know how malaria happens to us. Now, there have been different drugs that have been evolved, discovered to treat malaria. When I was a kid, I remember it was chloroquine. And I also remember some of the side effects of chloroquine, how I used to run away, you know. I at that time I preferred to even stay with malaria than to take chloroquine because I would take chloroquine, it would weaken me so much. I would have itching and all the likes, my eyes, you know, a lot of other things that will happen. Anyway, chloroquine used to be very effective when we were young. You know, they give you the injection and then you're better. Okay. But in no time, it's became useless. We still have some people that go to pharmacies and tell you that, oh, chloroquine is what they use. I smile a lot of times. Okay. It doesn't mean it is completely useless. It still retains a little bit of efficacy, but it's not something we can practically use to treat malaria again. The next time you buy chloroquine off the counter, just try to look at the indication written in the leaflet. You will find that malaria is no longer listed as one of the things that chloroquine should be used for. The mechanism of action is a very beautiful one. It inhibits the conversion of heme, okay, to the innate beta hematine. Now, heme, when the parasite breaks down red blood cell, of course, hemoglobin is digested. Hemoglobin is a massive protein that contains about, I think, about 4 units of the porphyrin ring called heme. Okay, so when the heme is dislodged from hemoglobin, the heme itself becomes very toxic to the parasite, to the plasmodium parasite. So the plasmodium parasite has found a way to render it harmless. And the way is by making sure that it converts the heme, the single heme molecules into like the crystal form of beta hematine, hemozoin, which is completely harmless. So what chloroquine does is to prevent the ability of the parasite to convert heme to hemozoin. So the moment the parasite is not able to do that, the heme that is freely around the parasite in the digestive vacuole kills the parasite. So, but with time, we started having widespread mutations in the efflux transporter of the Plasmodium falciparum chloroquine resistance transporter. And because of that, what it means is that by the time you take chloroquine is supposed to be concentrated in the food vacuole of, I mean, of the parasite. But what happens is that the parasite has figured out how to efflux, how to pump chloroquine outside. So it doesn't really reach a critical concentration needed to inhibit the conversion of heme to hemozoin. So because the parasite has developed that capability, chloroquine stopped having effect against it. Then we have the antifolates, the sulfadoxine pyrimethamine. If you're talking about Fansida, maybe the old schools among us, we know what I'm talking about when I say Fansida, because we no longer have Fansida in the market. The company has since been sold. So we have what we call Swida. Okay, you're talking about Fansida, you're talking about Swida, Amala, Maloxine, and the likes. They usually have two molecules inside them. The first one is sulfadoxine, which inhibits a particular enzyme in the pathway. The two of them, they work on the same biochemical pathway. And the second one is pyrimethamine. So they have this complementary activity. So one inhibits the first stage in the biochemical process. The second one inhibits the very next stage in the process. So by so doing, they are able to suppress the parasite and kill the parasite. But very soon, but bear in mind that we're talking about the use of two molecules already here at this level. At the time of chloroquine, it was purely a single molecule that was being used. But by the time you get to sulfadoxine, pyrimethamine, two molecules were being used. And even then, this parasite was able to develop high level resistance across Africa with point mutations. I mean, some of them are listed. The amino acid 108 converted from serine to asparagine, about three of them. And the parasite again was able to render useless this beautiful molecule. So we see use of adductin pyrimethamine, especially process, for what they call it now, the prophylaxis in pregnant women. Okay. And soon we started having the endoperoxides, artemether, artesunates, and their series. There are many of them. Artemether, you have artemether, you have artesunate, you have artemisinin, you have dihydroartemisinin, quite a number of them. So when these molecules were discovered, the first one being discovered by Tuyuyu in 1972, everybody was excited. This was a very beautiful molecule. Okay. The mechanism of action is that it's cleaved by the heme ion, the same heme that is generated from the degradation of hemoglobin. It now generates toxic reactive oxygen species from the molecule, which targets a particular ATP enzyme of the parasite and the catch 13, and the parasite dies. Highly toxic against the parasite. Everybody was happy. But because of what the scientific and the medical community they had learned from the previous drugs, chloroquine, sulfadoxine, pyrimethamine, that this parasite was able to develop resistance. So they made sure that these new molecules, new at that time, they were not used singly. They were not used as monotherapy. They made sure they were used together with other categories of anti-malaria drugs. For instance, artemether will be combined with lumefantrine, artemisinin with piperacin, and the likes. So they evolved the concept of ACT, artemisinin combination therapy, which everybody believed would be the solution to the penchant of this parasite to develop resistance. So I think generally when that happened, when ACT was released, the research community seemed to pause a little bit their discovery research because everybody believed that what we have is a golden bullet, that the parasite might not be able to develop resistance. Unfortunately, partial resistance has been recorded, especially in Southeast Asia and part of East Africa, and it's actually coming fast to Africa too. The reason why we may not be alarmed so much in Africa is, I think, because we don't have data. A lot of people will be able to give testimony that now, when they use the same drug they had been using before, the same ACT, maybe Lonat or whatever, I've seen a lot of people that even after the 3 days of therapy, they still did not feel completely better. Okay. I've seen people that it's only about 5 days after taking the full course of adult treatment, that they will start feeling some kind of improvement in their symptoms. In fact, quite a number of them will have to go take another anti-malaria drug before they are okay. So it's already in our community. It's already here in Nigeria, the resistance to ACT, but we don't have proper documentation to be able to know the extent of the problem on our hands. Of course, you also have primaquine and the likes, the 8-aminoquinolones, which were limited mostly because of the G6PD deficiency. So a lot of things have been done. For instance, vector control. Okay, you want to control the mosquito. There were times that the government we initiate this kind of program. You want to check everywhere where the mosquitoes would breed. You want to make sure that okay, you turn the water away, you cut the bush around you, you use insecticide treated net, you use raid and fleet and the likes. We've done all this, yet people still contract malaria. Malaria still kills people. I personally have come to think that Africa is too big for us to be able to eradicate mosquito. So we still don't have a choice. We may have to go back to pharmaceutical means, especially drugs, to be able to control. Okay. Of course, there's counterfeit medication problem too. Counterfeit drug doesn't mean it doesn't have the molecule, but usually it may not have the content, the amount of the active constituent you're supposed to have in the tablet may not be there. And when you expose the parasite to suboptimal dose of an anti-malaria drug, you are actively encouraging it to quickly develop resistance. So these are some of the things that have affected us. From what I've said so far, you can bear witness with me that malaria as a disease has defied the traditional discovery pipeline. On the screen, you will see the general drug discovery pipeline. If you want to discover a new drugs, these are some of the main stages that you have to go through, especially when you're talking about monotherapy or even some level of combination therapy. You start with the concept, you select the target, which is the receptor you're interested in, usually one receptor, and then you do validation of that. You find lead molecule. Lead molecule is a compound that is able to interact, bind with the target, and give you the kind of biological effect you are interested in. Of course, you do some level of optimization, maybe to improve pharmacokinetics or to make it patentable or for other commercial reasons. And then you go into living organism testing. The first stage is the preclinical development, which is mostly in animals. You go into clinical development, different phases of human trials, and then eventually, if everything goes well, okay, you get an approval to start selling, to get that marketing to larger populace, and you continue with marketing authorization or post-marketing surveillance, I mean. Okay, this has worked for so many drugs. It has worked for antihypertensive drugs. It has worked for anti-diabetic drugs, for a lot of drugs. But in the case of malaria, I don't think this pipeline is going to give us the kind of results that we desire. So there are many other ways that people are approaching this problem of malaria. The first one is development of vaccine. I'm sure many of us have heard about that, and also monoclonal antibodies. They are not really 100% efficacious, but it's worth trying, okay? And then we have the concept of polypharmacology. Now, as a pharmacist, I find this very interesting because when I was in school years ago, we used to snidely remark about polypharmacology. We used to talk about polypharmacology or polypharmacy as if it were something very bad. It's something that exposes a patient to multiple compounds at the same time, which can increase the possibility of side effect or even ADR, adverse drug reactions, and the likes. Okay. But in the case of malaria, you will see that even before now that we have systematically adopted polypharmacy, that is the use of multiple drugs for the same disease, like ACT, for instance. Okay, artemisinin plus lumefantrine or whatever. That's already some level of polypharmacology. But with the development of resistance against the two molecules we normally use for treating malaria these days, you will bear witness with me that two drugs may no longer work. And I'm here to prove to you that that may be the new path that some of us will have to follow up. Now, what is CADD? Drug design generally assumes that there is a little bit or a lot of rational effort in the discovery or design of new drugs. Historically, many of the drugs that we have on our shelves or that have solved our disease problems, they were either obtained by accident. There are many of them, serendipity, lucky accidents, like penicillin, like even morphine, like Viagra, sildenafil, many of them like that we're not actively looking for those molecules, but we found them all the same, or by just doing brute high throughput screening when we don't even understand the basis, I mean, for the activity that we are discovering in the compounds that are being discovered. Now, in the case of drug design, we are telling ourselves, we are telling the research community and the public that we are logically, rationally creating a new molecule because we understand exactly how the molecule works and what it should look like. So this actually predated computer-aided drug design. People had been putting one and two together, the biologists, the chemists of this world, they had been putting one and two together to understand that, oh, okay, this kind of molecule is the kind of molecule that can have this kind of effect, whereas this kind of molecule may not have this effect and things like that. So, but CADD now makes it easier to do. Okay. It makes it possible for the research community to also do it in a very fast way. So CADD integrates computational chemistry, biophysics, structural biology, and informatics to streamline the identification, design, and optimization of biologically active compounds. Okay, it reduces the time that we need to discover new lead molecules from 5 years to few months. And some of you probably you were aware of what happened during COVID-19, the speed at which the research community was able to respond to that pandemic. It had never happened in the history of mankind before. In fact, when COVID-19 happened, they had to start revisiting and revising textbooks on drug discovery. Because they will tell you that, oh, okay, it will take years before you can discover a new drug molecules. By the time COVID-19 was here, because we had access to computers, things were done within weeks, not even months now. Okay. So it's also able to reduce the cost. It saves millions of dollars literally by filtering out inactive compounds prior to wet lab synthesis and extraction. Some of us that are interested in natural product research, for instance, we don't start our research work again now just by going to collect plants. No, we don't do that. And a lot of times when you go to collect plants, you probably will be able to collect only one or two plants to study at once. You understand? And you are limited by cost, you are limited by time, by a lot of things. But now, working on CADD or with CADD, I might be able to screen hundreds of plants before I even enter the bush. I will eventually hit the bush, but before I hit the bush, I know exactly the kind of plants I'm looking for. Okay. And the beautiful thing about CADD is that it provides information about mechanism. So if you are starting your drug discovery project with CADD, you already have an idea, the target system, the enzyme, the receptor that your active components are expected to interact with, even before you go into in vitro or in vivo assay. Okay, so next slide. I'll just give a very, very general introduction about the core methodologies in CADD. And at the end of this, I'll probably go into a little bit some technical detail, but I would like to reduce them so that I don't bore us too much. Most people don't like equations and the likes. Okay, so we generally have structure-based drug design, in which case you have a three-dimensional structure of the receptor. Let me use that word, of the receptor molecule that particular compound you are looking for is expected to interact with. Okay, for instance, let's say I'm looking for a new antihypertensive drug and I want to explore the adrenergic receptor. Okay, so the adrenergic receptor has already been solved. The structure, okay, the chemical structure, or what we call the three-dimensional structure, has been solved by biologists, and the structure deposited and available freely. So where I'm starting with the structure of the receptor, we call it structure-based drug design. The structure here is talking about the structure of the receptor. And some of the methods we use, most of us here will probably know that it's molecular docking, which is used to predict docking ligand binding posts, okay, and the binding affinity. We score it so that we can prioritize different posts and know which one is the best to work with. And a very powerful method also under SBDD is molecular dynamics simulation, which I'm sure some of us must have also come across or have even used. So we also have ligand-based drug design, which utilizes pharmacophore mapping and quantitative structure activity relationship modeling. Okay, there are many other approaches under both of them, but these are the most common ones. I must also tell you that the structure-based drug design is commoner compared to ligand-based drug design. Okay, I'm just limiting myself to SBDD, the structure-based drug design now, and giving us a general format of what the equation that we solve when we're doing MD simulation, what it looks like. When MD simulation is based on the computation of potential energy functions, whereas the second one talks to docking, where we are computing the what we call affinity, but affinity, the word affinity is actually a misnomer. The right word is free energy of binding that we compute, usually computed from the enthalpy, which is mostly the potential energy between the interacting partners, and then the entropy. Okay, entropy is usually computed from how do I say it now, from solvent system. Okay, but usually most times we don't even include solvent system. By the time we are doing docking, we just assume a particular dielectric constant for solvent. In any case, you can also see the relationship between the binding free energy delta G bind and the computed inhibitory constants, okay, Ki minus RT lane. If you get a particular binding free energy, if you want to understand what the inhibition constant, the experimentally obtained inhibition constant is supposed to look like in a pure system where you have your your ligand and you have your receptor, you can use that equation and it will give you a very good approximation of what it will look like. Now, if I start talking a little bit more about docking, what do we do in docking? We have our receptor system. You can, if you look at the right hand of this slide, the whitish surface bulb is the receptor. I've simply just colored it with white. And then the position where the ligand is supposed to bind to, I've put a box, a colorful box there. One dimension is colored red. I think that should be the X dimension, and the Y dimension is colored green. The top dimension is the Z dimension is colored blue. Okay, I centralize that grid. We call it grid, this grid box. Okay, and then I now position sequentially my ligand, okay, or ligand system inside that box and compute how that ligand is going to interact with the receptor molecule. Usually, when we do that, we fix the coordinates of all the atoms of at the binding site of the receptor and even all the atoms of the receptor. So we call that rigid docking, okay? And our ligand is the only one that is geometrically optimized. Okay, somebody's asking a question. I think we may have to take a look after this talk. Okay, so it's only the ligand poses that are geometrically optimized, especially the torsion, the dihedras in rigid docking. That's what most of us do because it is cheap computationally. It's also cheap temporally when it comes to time that we need to get the value that we are looking for. It's usually very beautiful when we use it for a very large data set. But by the time you have just maybe one or two compounds that you want to dock, it's more advisable to go for the second one that we call flexible docking. So flexible docking introduces some level of flexibility to the binding sites of the receptor and also the ligand. And many of the docking app that we have software, they are increasingly capable of doing flexible docking. But the best way to introduce flexibility is by using molecular dynamic stimulation. But before I go into that, I presented the same receptor. This is acetylcholine binding protein from a mollusk. Okay, that's the name of the mollusk. You can see image one, that's A and B. They're actually experimentally determined by X-ray crystallography. You can see that the same receptor actually fundamentally has different shape at the binding site. A is the closed conformation and B is the open conformation. You can see nicotine band to it. And you can also see the what it looks like if we are using the new cartoon, what we call the ribbon. You can see the opening of that particular loop. So this happens normally in our living system, in our body system. Nothing is actually static in the body. So a lot of the docking that we do, we are just oversimplifying a biological reality that encodes so much flexibility. Okay, so some of the two quotations that the entire field of molecular dynamics simulation is premised on. The first one is from Richard Feynman, who won the Nobel Prize in 1965. And basically, he said, if you were to name the most powerful assumption of all, which leads one on and on in an attempt to understand life. We're talking about life now. We're talking about living systems. Okay, it is that all things are made of atoms. Okay, and that everything that living things do, whether we do it as on a macroorganism level or inside our body, everything that living things do can be understood in terms of the jigglings and the wigglings of atoms. This looks like a joke, but it was speaking to the fact that every atom in our body, in our receptor, in the aqueous system of our bloodstream is always moving, vibrating about a particular main condition, main position. And when the environment changes, maybe temperature changes, maybe isotonicity changes, maybe the pH changes, these affect all these winglings and jinglings. So the next quote is from Pierre-Simon Laplace on causal determinism. Some people will call it the Laplace demon. So he said, given for an instant, I like this particular quotation, given an instant and intelligence. In the place of intelligence, you can just put computer, okay? You can put our brain too, but you can also put computer, which could comprehend all the forces by which nature is animated. when you're talking about animation, you're talking about motion, okay? It's basically saying that we have a system, we have a computer that can audit all the forces acting on an atom or a molecule or on a receptor and the respective situation of the beings who compose it. An intelligence sufficiently vast to submit this data, because it's going to be a large data to analysis, it would embrace in the same formula the movement of the greatest bodies of the universe and those of the lightest atom. And for it, nothing will be uncertain. And the future, as the past, will be present in its eyes. Now, what it means here is that you can predict exactly where what atom will be at time what, at the next time, at the future time. So if I take an atom now, for instance, and I understand exactly the forces acting on it, either the force acting from within it, if that's possible, or it's acting from outside its environment, I can be able to predict that, oh, okay, 5 minutes from now, this will be the position of that atom. 5 years from now, this will be the position of that same atom. Okay. So, and this same thinking had been used for a number of things in prediction of solar eclipse and also now in MD simulation to be able to predict where what atom would be at whatever time. If you look at the time, the date when this man authored this word, when he authored these words, it was in the 19th century, 1812 to 1820, long before anybody knew there would be a computer. But the intelligence he was talking about, because nobody could comprehend that there will be a platform, there will be a machine that will be able to do all this calculation at that time. In fact, what he said, they used to call it Laplace demon. There's only a demon, a spirit being that will be able to do that. But now we have computers that achieve all this. Okay, if you now go to what MD simulation looks like, I won't dwell so much on that. We've already taken some time. Okay, we audit the forces and then you predict what where it will be. Okay, when you calculate the force, you usually don't calculate forces, we calculate the potential energy. And when we take the second derivative of that, we can get the force back, the negative derivative, by the way, we get the force back. And from the force, we can also go back and then we calculate where position will be. And this is the general equation that is used. This equation looks complicated, but it's very simple. The bonded terms in any molecule, they are computed with the three expressions on the left, and the non-bonded, we're talking about electrostatics. You can see at the far right, you can see the formula, the equation for electrostatic computation, QI, QJ. That's just the specific, how do I say, the partial atomic charges between two atoms and the likes. Very simple. This is what all your MD simulation packages, Chromax, Amba, NAMD, Charm, this is the equation that all of them solve to be able to give you an information about the dynamics of your system. Now, CADD has been used to discover real drugs. I remember years ago when I was doing my master's in London, the master was in drug discovery, and I was about to choose which area I would do my research in. When I left Nigeria, I wanted to be in synthetic chemistry. But so my first time of coming across CADD in 2008 then was in London. I was not sure because I thought it was too good to be true that we could use computer and calculations to discover new drugs. So one of the things that helped me in making that decision was the realization that pharmaceutical industries, okay, they were using the tools at that time. I know pharmaceutical industries don't play around. They don't do theoretical research. They do realistic, practical research. Anything pharmaceutical industries are doing is to solve problems. So we paid a visit to GSK, I think in Stevenage, Northern London, and I saw some of the computers I'd never at that time seen in my life that are not even available on the market. And by the time I started reading the history of CADD and then trying to like understand whether they have been able to produce drugs from using this method, I was impressed and I was pleasantly surprised. Captopril, for instance, okay, is a drug that we use for hypertension. Indinava is for HIV. And then we have Tamiflu and especially imatinib. At the time when I was doing my master's, imatinib was still the hot cake. I think the patent had not expired at the time, so almost every lecture they gave us in the school would include Glivec. Where this slide is telling you that CADD is being increasingly leveraged to study and discover actual drugs in the market. Okay, these are some of the research work I've done using CADD I'm talking about real drug discovery research. We're not going to spend too much time. I'm just trying to make a case for CADD and how we can leverage on that in our rethinking strategy for discovering new anti-malaria drugs. This was the work we did on COVID-19. And like I said, within few weeks, I was the one that personally handled this particular project. We're able to screen more than 1.2 million compounds, against the SARS-CoV-2 main protease enzyme. And then by the time we were done, we got we wouldn't, of course, we wouldn't go through 1.2 million compounds against the receptor one by one. That's impossible to do. Even though we did not use machine learning or AI or something like that, but there were computer programs, especially Python scripts that I could write to do the analysis for me. Okay, so and then we streamlined it to a few of them, and I performed MD simulation, hundreds of 100 copies of 100 nanosecond MD simulation. And based on that, we're able to choose the best compounds. We ordered them, we subjected them to in vitro enzyme assay, and we got very interesting results. Okay. Now, this is the in vitro results. It was, I was personally impressed that we could transit from computer to real experimental activity. But we didn't follow up with the research work because we did not believe that COVID-19 was a disease or was an infection that was going to stay around for so long. So there's no point spending too much money trying to discover a treatment for something that will soon leave. Okay, now we're coming back to plants. I am a very good, how do I say it now, fan of getting drug molecules, new leads from plants. Now, almost everybody in Nigeria, in Africa generally, is working on plants, whether you are in biochemistry, in animal science, in pharmacy, in medicine, one way or the other, we all work on plants. But the common approach that has been used is what we call ethnomedicine. Ethnomedicine relies on ethnic knowledge, okay, on what our forefathers or ancestors had known about these plants. Maybe they are using Azadirachta indica, that's the name. Maybe they're using it to treat fever. So it makes sense for us to think it may be useful in malaria, okay? So as a scientist, what do? You go back to the plants, to the herbal potions that our forefathers used to do. You subject them to experimental approaches and see whether we could validate those particular expectations from those plants. That's what we've been doing mostly, okay? This is beautiful. This approach had already given us a number of drugs. We had morphine from plants. We have quinine, digoxin, ephedrine. But interestingly, in the field of malaria, we have artemisinin. This was how artemisinin was discovered. In 1967, there was a plant screening campaign under a secret military program codenamed Project 523. It was set up by the People's Liberation Army to find an adequate treatment for malaria. 1967, look at that date. The program and early clinical work were ordered by Mao Zedong to provide assistance to their malaria-ridden army. So this was a military project, okay? Malaria was killing a lot of their armies, but so they wanted their own drug that would prevent their soldiers from dying. So in the course of this research in 1972, Tuyuyu discovered artemisinin in the leaves of Artemisia annua. It was one of the many candidates tested as possible treatment for malaria by Chinese scientists from a list of nearly 5,000 traditional medicines. This is ethnomedicine. Now, imagine how much that would have cost. Imagine how much it will cost to gather about thousands of plants, or maybe plant potions. I don't know whether they tested them singly or in combination, okay, in Agbo fashion. Imagine how much that would cost. I mean, some of us that do extraction, you can already start guessing how much of solvent they will have to buy, how much of this they will have to do. They will spend literally millions even at that time. So eventually, this woman discovered these plants. And I can tell you that was not the only plant that she discovered, but the most active at the time was Chingaozu. That's what we call Artemisia annua. So that's ethnomedicine. So ethnomedicine relies on the back of ethnic knowledge, the experiential knowledge unique to an ethnic group. The Yorubas, we have our own. If you go to the Igbos in the southeast, they have theirs. The Northerners, they have plants that they peculiarly use to treat different diseases. This is what we call ethnomedicine. So there's a reliance on that ethnic knowledge in deciding the plants to turn to in the search for new drugs. So we as a scientist, if I'm looking for a new anti-diabetic drug, the first place I may start from, if I'm doing ethnomedicinal research, is probably to go and carry out a survey, talk to babalawos, talk to how do I call them now? Herbalists, and then try to get information from them. So from the information they are giving me, I may now go into the lab, collect the plant, and see whether it will have that effect. So this indeed worked magically. However, I must say that we have roughly about 400,000 vascular plant species on the planet. And we humans have only historically had experience with a tiny fragment of them. And oftentimes, that experience is in limited use context. So out of the 400,000 plant species thereabout, and new ones have been identified, by the way, every year. So out of the 400,000, perhaps we have not been able to go through even 10,000. Now, imagine the amount of therapeutic potentials that we have not been able to check out. And the reason is because we have limited ourselves to what our ancestors knew. For instance, if a plant is present in Kano, let's say it's naturalized, it's only present in Kano, it's not present in Oyo State. So there is no way a herbalist, no matter how good he is, in Oyo State, there's no way he would have had experience with that plant that's only present in Kano, unless it travels. Okay? Now, it is also possible that there's a disease in Oyo State that is not in Kano. Let's assume there's a disease F in Oyo State that the people of Kano do not know. And it's also possible that it's only the plants in Kano that can do the work best, that can treat people against that particular disease. So as long as you are separated by distance and experience and knowledge, we have a very big weakness for ethnomedicine. Now, there was some group years ago that tried to experiment how effective effective ethnomedicine could be. The National Cancer Institute, they had this anti-cancer screening program, and they got this in their work. They screened random samples for anti-cancer properties in these particular compounds. By doing random screening without any kind of information of whether it has some effects against cancer or cytotoxic effect at all, just randomly screened compounds, they were able to get about 10.4% hit rate. Hit rate means compounds that show interesting activity, okay? So when they use ethnomedicine, they were able to get 19.9%. So it means the decision to include a particular compound or particular plants relied heavily on information they got from ethnic practitioners of herbal medicine, okay? So they're able to get 19.9. That's almost double the hit rate they got by random sampling, okay? But it's also a little bit disappointing, though, that it's not more than that, right? Now, by the time they screened poisonous plants, they got a hit rate of 50%. I'm sure people and farmers in this place already understand why that might be so. Now, poisonous plants are normally avoided by ethnomedicinal practitioners. If you're a herbalist and you find a plant to be poisonous, you're not likely going to include it in your agbo. It's almost like including a plant like Datura metel, okay? In agbo, something you need can kill people. You avoid it. But interestingly, poisonous plants are actually highly pharmacologically active at low doses. It makes sense to even start looking at them for potential drug action. But ethnomedicine runs away from them. So this is one of the major weaknesses of ethnomedicine. Okay, now the second study by the Centre for Drug Research Institute in Lucknow in India, they also did random screening of plants. They got 18.9%. This is very impressive, just random. And when they used plants with associated ethnopharmacological uses, the hit rate was just about 18.3%. So there is basically no difference between the random screening and ethnomedicinal screening. This doesn't mean ethnomedicine is not useful, it's massively useful. But when we're talking about malaria research, it may not be able to give us what we are looking for. Now, the questions we need to ask, are there other disease areas where the same preparation extra natural products could be useful and possibly even more useful. Let's say you are using Azadirachta indica for fever, for instance. Is it possible that that same plant that you're using for fever, okay, could have been much more useful for asthma? But ethnic knowledge, ethno-medicinal knowledge may lock you into Azadirachta into febrile treatment alone. Okay. Question 2, are there other natural products present in the plants whose pharmacological potentials go unnoticed because they are only present at a very low dose? It is only when you have that information that you may have to like tweak your extraction science to be able to concentrate those compounds that are lowly expressed. Okay. So in ethnomedicine, is the predominant plants, the ones that are concentrated the most in your extraction, that you feel their effect. The minor ones, you may not feel their effect, however beautiful those effects could potentially be. Question 3, is it possible that potentially valuable natural products are missed on account of pharmacokinetic exclusion? For instance, if you have a particular natural product, let's say flavonoids, that are not well absorbable, the bioavailability is so low. If you have to use it for a systemic infection, a systemic disease, you may not be able to get the benefit of their activity. Why? Because they don't even get absorbed. But the fact they don't get absorbed doesn't mean they are useless. There's a way you can do optimization to be able to get them to be useful. Okay. So the last one is it possible to cut down on the cost? Yes, it is possible. So considering the large number of plant species that we have, and the fact that we have a little bit of limitation with ethno-medicinal knowledge, anti-malaria lead discovery effort in Africa will require a lot more than dependence on ethno-medicine. Okay. Often we have examined the ethno-medicinal extract, like I said, in very narrow disease context. Our solutions are doubtless in our plants. I personally believe that the solution to malaria is in the plants that we have in Africa, mostly. Maybe we're not just looking, in the right places yet. So we additionally have to adopt a scientific strategy that accommodates clinically relevant features of the malaria pathogen. Features like its ability to develop resistance. Features like the kind of morbidity it can cause in the body. Features like the complexity of its life cycle. Most of the drugs that are available, they only treat a particular phase of the life cycle of that particular parasite. So in Africa, of course, I don't have to go through this. We know that we are rich when it comes to biodiversity. We have so many plants. And these plants, we've had alkaloids, terpenoids, flavonoids, phenolics that have shown this effect, at least in vitro assays. Okay, so we are not in want of possible plants to turn to. Now, I will quickly go through the project, our anti-malaria project that we have been working on for at least upward of 5 years right now. So this project has been conducted in 2 phases. The first phase, what we basically did was to build a natural product library of over 3,000 from over 200 Nigerian plants. We simply took one plant at a time and identified all the natural products that have been reported in each plant. Then we generated the 3-dimensional structure. We made sure everything was okay. The connectivities were fine and all that. We did energy minimization. We stored them in PDB format, PDB-QT format. So the next is that now we want to search for possible, let's say, potential anti-malaria properties of those plants. And we don't want to enter the lab yet. It's going to be very expensive to have to collect 200 plants and make their extract, and then start to check whether they are going to have the activity we need, maybe in vitro assay or in vivo assay. That would be too expensive. And I don't even think any grant awarding body is going to give grant for that. It's almost like a waste, especially because we have technological options that can reduce that in our age right now. So what did we do? We tried to search information about some of the critical macromolecular targets, let me say enzymes of Plasmodium falciparum, that if we're able to inhibit them, we may be able to disrupt its life cycle, its biochemical process, and also kill or inhibit the parasite. Okay, so we came up with 8 such targets. The first one is alanine aminopeptidase, and the next one is peptide prolysis trans isomerase, and then poly-tRNA synthetase. Now, what we did for this first one was to screen the 3,200 plus natural products against the three-dimensional structure, X-ray crystallographic soft structure of each of these serially. Serially means we started with number 1 enzyme. We screened all the compounds against. Then we selected those of the compounds that were able to outperform the co-crystallized inhibitor. Okay. Then we passed those through the of how we did the docking. This was just purely docking, no MD simulation at all. And for a very good reason. The reason we didn't do MD simulation was because we're not looking for natural products. We're not looking for pure compounds. We're not trying to discover pure chemicals, okay, like conventional drugs that we have. We are trying to identify the best plants to combine to be able to develop a herb-based treatment. So that's why we didn't have to do MD simulation, okay, because the focus is not on individual phytochemicals. So if you look at the scheme, we started with 3,212 natural products screened against, the first enzyme. So the best from that particular screening was 861 screened against the next. So the best from that was 691 screened against. So the best from the third, there were 172 natural products. So that is the end of the serial docking. And you can also how do I say, appreciate the fact that the 172 that we got from the third enzyme, each one of them must have done well against the 2 previous enzymes. Okay, so every single natural product, every single one of the 172 natural products has the ability to bind those three enzymes. So we can say that we have compounds able to interact with multiple enzymes of Plasmodium falciparum. So at that level, we now transited to parallel screening. So what it means is that the 172, we screened against the 5 enzyme panels, one by one. So at the end of the day, when we got the results, we simply obtained an average, okay? And then we rank them, the best compounds out of them. We gather them together, and those best compounds we now look back at our library, natural product library, to identify 20 Nigerian plants that contained those natural products. Okay, that contain most of them. And each of those plants identified contain multiples, okay, multiple such natural products. So we collected them, we subjected them to beta-hematine assay, that's in vitro assay, and then we obtained the results. Okay, I've encoded the plant name here. So the chloroquine was used as the standard. The IC50, the best IC50 was gotten for one of the plants, which was actually superior to the value we got for chloroquine at crude level, which we consider to be very significant, by the way, because at crude level, we're supposed to have just these natural products present at relatively lower level than the chloroquine used as a pure compound. Okay, so so these are the top 4 plants, and you can see the results, the IC50 results obtained from there. So this was not the end of the work. So what we now did next, we now went back to the drawing board. We went back to those plants. Because some scientists had worked, and there are new, let me say, new publications of new natural products in those plants. Okay, so we basically rebuilt our phytochemical library for those 20 plants. From those 20 plants alone, we had about almost 950 phytochemicals. Next, we now said, okay, we're not satisfied with just using 8 targets of malaria, of Plasmodium falciparum. We escalated to 72. So we collected the 3-dimensional structure for 72 of such targets. We prepared them for docking, and we commenced the massive docking. Now, the 76 targets, they were not unique necessarily. Some of them, they had redundancies, okay? It means, let's say, for instance, now the PFK2, we might have up to about 5 different extra crystal structure for that one alone. But we included all of them because we also know that by including multiple conformations of the same enzyme, you're actually incorporating flexibility in your study. We call it an ensemble approach to docking. But the 76 targets that we used, the 76 PDB files that we used, we had 24 unique target systems in them. And these are the ones that have been listed. Okay. Of the 24, of the 76 also, 5 of them were actually resistant variants. So which we expect might have been resistant to a certain form of compound that they may have designed against them. So we also included them. And that's because we're expecting that whatever product we bring out should have effect against even resistant varieties. And then 8 core functional biochemical mechanisms were targeted across. Okay, the pyrimidine and purine biosynthesis salvage, the glycolysis, the fatty acid biosynthesis, and so on and so forth. So we want to be confident that by the time we have our product, the product will be able to work across multiple mechanisms at the same time in the same product. So but before we go on, the question is that if we are indeed able to bring out a product that can achieve 100% simultaneous inhibition across all the life cycles implicated in this study, what would be the effect? The effect for the parasite will be catastrophic. That's one. And we believe it's going to be able to attack multiple stages of the life cycle of that particular parasite. And what would be the effect on resistance? We think it will make resistance virtually impossible. And that's because if a single point resistance arises, the probability is about 10 raised to the power minus 9. If we now target 24 different independent genes and biochemical systems, that's about 10 raised to the power of minus 216, which is more than the number of all the total parasite population that we have. So it is virtually impossible for a parasite to be able to develop resistance to such a treatment. This is the ideal. This is the hypothesis, okay? It doesn't mean this will be the reality. The reality will come down to by the time you have such a product, how do you ensure that the natural products implicated that we are showing computationally to be able to inhibit these compounds, how are we sure that they will be in the final product and at the right amount? Because we all know that we have this problem with herbal medicine standardization, but that will be the problem. That will be a challenge for the later time, okay? So we redid our extraction now using ethanolic extract. We did in vivo testing and we got some fantastic results individually in the best plants. The same plants that we showed the in vitro results, this is now the in vivo, part of the in vivo results. We are not done with the full analysis. Maybe in the next few weeks to months, the full picture will come out and we'll be able to submit for publication. Some of the plants have some of these are some of the compounds we find in them that were able to strongly interact with multiple primary targets of Plasmodium falciparum. And this is the general interaction matrix to show the activity of even some of the ones that were not shown there, that how do I say, interact across different target panel. I think at this stage, we have been able to choose plants that contain natural products that are able to bind to multiple targets. And then we are confident, we're not so confident, but we believe that some of the effect that we are seeing in vitro and in vivo are actually a response, or rather a consequence of the binding studies that we have done, although we still need to do a lot more work to be able to escalate that. Okay, one of the things that we may have to do next is we are going to have to do the toxicological assay to see how safe it will be for humans to take these plants together, okay, and whether it will be safe for which category of people. This will be all animal study. So in rethinking drug discovery strategies in Africa, we have to start thinking of polypharmacology. And the best way to get polypharmacology is from nature. Every single plant that we are holding is literally a laboratory of chemical compounds. I mean, I think a very modest estimate puts it at about that in a plant you have more than 100 chemical compounds. Okay, so there is no better way to have polypharmacy than turning to plants at this time. But we're not going to do it the way it has been done. We have to do it intelligently to be able to make sure that whatever we are bringing out is actually something that will knock out the parasite for good. So for we that are young researchers, we have a lot of options. I know somebody is probably asking a question about AI and then machine learning. Yes, these tools are there, but the danger in them is that they make things so easy that they shield us from the basic science, from the core science. So you have a lot of scientists now who are not well grounded in their field, and that's because anything you plug on AI or before you know it, it brings the solution out. And you simply just present and everybody thinks you're smart. But the science behind it is shielding us from it. So we should be very careful when we're using these tools. Let's take the pain. Let's make the effort to understand the science, the calculation, the equation, and how to interact at the machine level. Then we have to encourage cross-disciplinary research. One person cannot do everything. The fact is that the work I've presented today, there are many of us that worked on it. Okay, some work on extraction, some work on in vitro, some work on in vivo, and we have a lot of students that were supervised, during the course of this project, which is still ongoing. And I also want to sound the note of warning to us that let us always endeavor to validate our in silico data. When you have made predictions in computer, you use AI or whatever, the best that result is called a prediction. Okay, it's a prediction. It doesn't become an activity statement until we have developed one way or the other to validate our results. So I want to thank us for sitting through this presentation without sleeping too much. I thank Dr. Wande, Pam Shedrack, and everybody that has made this to be possible. I also thank my research students and all the researchers that we have worked with. I should have, if I had more time, I would have put all their faces and their names here. So thank you very much. That's the end. So if we have questions, I don't know how you plan to coordinate the question, whether they will drop it in the box or people will have opportunity of calling in.
Yeah.
I'm here.
Yeah. All right. Thank you so much. Thank you so much, Professor. I don't know. I really appreciate it. I know everyone too enjoyed the class and yeah, it's great. So I think it's time for question. If you have a question, we'll start from maybe asking general questions. You can indicate with a raise of hand. Then we can take that, and after that, we'll go back to the chats. I don't know in case I also personally, I dropped some questions in the chat. So, but we can start from a raise of hand. Okay, Catherine, I think you can unmute yourself and then ask your question. Yeah, Catherine, I think you can unmute yourself. You'll be the first person to. Yeah.
Good evening, sir. Thank you for the very enlightening presentation, and a very big thank you to the organizers. Thank you, sir. I have two questions. One is a technical question and one is a general question, sir. So my technical question is about the free energy or potential energy calculation. So for the non-bonded interactions, there are electrostatic, van der Waals, and some will also add hydrogen bonding. But there's quite a debate about whether or not the van der Waals interaction is a long range or short range interaction, and I just want to know what your view is on that, sir. Then my second question is about the drug design process from research to market. So you mentioned that the goal of the malaria research is to get compounds that are active from the natural product library, and then the end product is meant to be an herbal medicine. But I have a question. If at the end of the day the herbal medicine is very effective, with the population of malaria-infected citizens in Africa, it means there's going to be a rise in the sales, and you will need a lot of herbal products to meet the demand. Do you What's the role of synthesis in that mix? Because you think producing herbal medicine to be sustainable, especially in this world of green chemistry and all that. What do you think is the role of synthesis in that, sir? Those are my questions. Thank you, sir.
Okay.
Thank you, Catherine. So, Professor, I think you will take them one after the other. Yeah.
Okay, let me start with the general question. But before I do that, I think I know that voice. That's Catherine Babalola, right?
Yes, sir.
Oh, it's good to hear your voice.
Thank you,
That's
sir.
okay. Now, how do we handle the ready availability of herbal products? That's the question, right? And what's the role of synthesis in that?
Yes, yes, sir.
Now, what is synthesis? Synthesis is creation of molecules in the laboratory. Is that correct?
Yes, sir.
I know you're a chemist. Yes, you create molecules in the laboratory, but that is happening in the nature every single second. Every plant is creating its own molecules, it's synthesizing its own molecule. So, what is the need for duplication of that effort when nature does it brilliantly? Now, nature doesn't just do it brilliantly, it does it in a way that an organic chemist is envious. Because nature creates molecules that you can never think of in a million years as an organic chemist. The complexity, the beauty in what nature produces, synthesizes is amazing. Okay. So generally, synthesis comes in one, if the plant sources, okay, will be problematic. You understand? But now, one of the things I've not talked about is the fact that this particular research project, we are making sure that we are limiting it to only leaves. There is no point of this project that we have looked at stem, bark, or root, because these particular parts of plants are not sustainable. You cannot commercialize them. By the time you have to use root to solve a particular health problem, how many plants do you have to cut their root and kill? But when it's leaves, you can actually grow plants sustainably and harvest their leaves. So we're focusing our attention exclusively on leaves. And now, whether they will be sustainable or not, I think it will be, we can plant them sustainably. We can. Even right now, we have a lot of natural products, or let me say, herbal products in pharmacies. Okay. How are they making them? How are they ensuring that, okay, they are not scarce? Why are they not using synthesis to try to solve that problem? Because those plants can be grown sustainably. I don't know whether that may not make you may not be very happy with that answer, because I know as a synthetic chemist, you want to find a way to how we can use synthesis to maybe solve the availability problem. But right now we don't even have the availability problem. We don't have it yet. In fact, right now we don't even have a product. It is when we have products that we have to start thinking about that availability problem. But even for synthetic products, I must tell you that at the time of doing drug discovery research, when you are doing the lead discovery and development, you are still not solving the problem of how to make it massively available. You are not even so much bothered about that. So by the time you now find out that that particular compound has become useful, has become a drug candidate, or maybe even an approved drug, that's when you start thinking on how to come up with synthetic methods to easily produce them. But I must also say again that we're actually trying to walk back from single molecule synthesis to something much more complex. We have seen that malaria challenge will not respond to synthesis, because how many compounds do we? I mentioned the fact that a plant contains maybe more than 100 natural products, 100 compounds. So how many of such can a synthetic chemist produce? And we're not talking about just a single plant. We probably will have to gather about 2, 3, 4, 5 plants together. Do you understand what I'm talking about? So
Yes.
the synthetic challenge may not arise because we are focusing on plants. I've not included their name for a very good reason, okay? But we are focusing on plants that can be grown, planted sustainably in Africa. I don't think it's a bad thing if all the entire Africa have to come to Nigeria and get this product that is in Nigeria. I don't think it's a bad thing for us. Economically, it's good for the country. Okay. Now, the question of free energy, potential energy calculation, the equations, the non-bonded parts, van der Waals, and how do I? You said electrostatic. You now ask the question whether the van der Waals is treated as long range or short range. Did I get that right?
Yes, sir.
Okay. Now, that debate is theoretical. That's the first thing. When it comes to implementation of these parameters, okay, I think parameter is the right word, either in MD simulation or any other computational platform, what we do in the implementation is completely different. It's sometimes, let me say, completely different from theory. Okay, and that's because we're trying to get the formalism, we're trying to get the form of that particular equation that can be computed. And you also notice that when you are doing MD simulation, they do things, you use things like cutoffs, right? I'm sure you've heard about cutoffs.
Yes,
Now,
sir.
cutoffs are artificial. They don't exist in nature. Cutoff is a way of saying, okay, I only want to compute van der Waals up to this point. I'm going to ignore beyond that point. No, doesn't mean the van der Waals forces are also don't exist beyond the cutoff. They do. But we know that the higher the cutoff, the greater the computational expense. A very tiny system, you may not be able to finish simulating it in 1 year if you don't truncate that particular cutoff at one point or the other. So when it comes to execution, it's not really so much of a problem. Some cutoffs, they have a switch function. Okay, they can say, okay, I'm going to truncate the computation of van der Waals from 0.9 Armstrong, and I'm going to, gradually bring it to 0 by the time I get to 1.2 Armstrong. So this will have additional computational costs to you. So, but it depends, eventually it depends on the level of accuracy you are looking for in your study. And when it comes to CADD, when it comes to drug design, docking, we don't need such accuracy when you are screening like millions of compounds. Okay, you just need something that is rough, a brute way of filtering them. So that's how it is done in CADD. We use cutoffs. And the fact that we use cutoff, it means we agree that these non-bonded forces, they can actually extend beyond that cutoff, but within the context of the study of what we are studying, we are limiting it there. Do you understand?
Yes, sir. I do, sir.
You're welcome.
Thank you, sir.
Nice to see you again, or to hear you again.
Yes, sir. Thank you, sir.
Thank you, Professor. So I think we'll move ahead straight. Chikani, I think it's your turn. You can unmute yourself and ask your question.
Hello, good evening, everyone. Good evening, Prof.
Please, can you speak up?
I cannot hear you. Please speak up.
Okay, good evening, everybody. Can you hear me clearly now?
Yes.
Yeah, we can hear you now.
Okay. Please, from the basic things I know about computer-aided drug design is actually trying to find the innovative molecules or compounds that are actually going to bind more to the target protein and probably achieve a better effect than the existing molecules we have now that are formulated in drugs. But then I reasoned that instead of taking this approach for malaria, can we know that malaria is not actually caused by one species of parasite, and even the same parasite can change genetically over time. And that's one of the reasons why the resistance to malaria drugs keep developing. So I'm wondering, instead of designing a drug only for the parasite as it exists today, can we use a computer analysis of the parasite's genetic information to predict the changes it could potentially make in the future, and then design a drug that can still work even when those changes occur? Because I believe that there is an advancement in artificial intelligence now and the machine learning, and those are fast computers that can help us in so many analyses. Even though there are different malaria causing plasmodium species like the falciparum, malaria, ovivas, and rest of them, we could still compare their genetic information using the help of those models and algorithms, and then find parts of the parasites that are similar across the different species, and then we'll now use the compared drug design to develop compounds that could potentially work against the several species at the same time, instead of just being so specific about the target protein, and then each of the compounds are now going to find more to achieve a better effect, because definitely mutations are going to occur in the future, and then we need to be proactive in order to find out a measure that we can use to curb the future occurrence. That's what actually I'm thinking. I don't know if it's something that is feasible or realistic. Thank you, sir.
Thank you very much. That sounds very, very interesting. I must say, it sounds beautiful too. And I honestly wish we could do that also. But what we are doing is also something in line with that. We're not just we're not interested in tracking the mutations the parasites may have tomorrow. We are interested in eliminating it so that it doesn't even get to that tomorrow. Now, let me quickly talk about how that particular mutation will happen. It's not as if you predict where mutation is coming from. The genetic process has a way of going from, DNA and nucleic acid walk to what they call the central dogma, which culminates in the synthesis of proteins. Okay, the proteins, the enzymes are what we are looking at. Okay, we're looking at the proteomic level, not really at the genetic level. So whatever changes, whatever mutations you have in the genetic level eventually comes down to the protein level. Now, what we are interested in is killing the parasite. There's a slide that I showed that showed the possibility or the probability of mutation occurring. We are not tracking the mutation. We are trying to make sure that whatever mutation it brings, okay, our product can eliminate it. You understand? Our product can eliminate that parasite. Not really the mutation is what we are interested in. Of course, it is possible. And then I also want to point out the fact that you said, we have AI, we have all these technological advances that can do all these things. I think we should be very careful not to expect too much from AI. And I know why I'm saying that. Human beings generally, when a new technology comes out, many of us talk about it. Many of us expect it to be the miracle that we think it is. You find out that a lot of times, even people who are not expert are even the one talking too much about it. Okay, I remember when CDD started, it was a big thing. It was exactly like the AI that we're talking about. Oh, it's going to make us develop all the diseases, drugs to all the diseases. This is the end of this and that. Diseases are nowhere to be found again. We're going to have drugs for everything. The fact is that people have received Nobel Prize in CADD. People have done a lot of things, yet we still have diseases. We have illnesses. So as long as the life exists, AI or not, AI will not solve the problem. AI is just a tool. Okay, it helps you. It helps us to do monstrous amounts of calculation. But I was sending a note of warning the other time that if you don't understand the science behind all these calculations, everything AI is telling us will be lost on us. So you won't understand what it's telling you. Okay, AI doesn't produce solutions so much like that. It simply aggregates data and brings pattern out that you might miss before. So, of course, what some of the things that I've done in this work is because I have access to AI and machine learning and the likes. You understand? If it were to be about 10, 20 years ago, it would take me much longer to do some of this work. Because by the time we are doing docking, we're talking about almost 950 different chemicals against 76 targets. How do you think we analyze all that? Because we have AI. But it's because we understand the core science behind it. That's why we can make sense of what we are getting. Otherwise, everything will be gibberish. Okay. So I think the approach I've talked about is right, but we cannot abandon the discovery of new products in search of that, which is highly still theoretical. Because in the present, malaria is killing a child every 1 minute. Thank you very much.
Okay, thank you, sir. Please let me just quickly ask another thing. With respect to this thing you just said now, because we shouldn't over expect so much from artificial intelligence, because people that even hype it are not actually the ones that know the science behind it. So I also want to know, is there any transition program that actually pushes us towards trying to learn more about the basics of artificial intelligence so that we that have the basic knowledge of what we want to do can be on the background trying to find out how it actually comes about. Instead of just being there to consume the product, we can actually go to the background and design it the way we want to be, and it can actually give us what we want. Is it possible?
No, of course, I believe there are so many programs. I've gone through a number of them, but I've just not found one that's giving me what I actually need. There are so many programs out there, but many of them are commercialized. But I always like free things. I think you just have to like look a little bit deep and you find the one that you want. Now, whatever you are going to identify or use must be something that solves your own problem. What problem I will have in my research will definitely be different from yours. Okay. So there are programs, there are different tutorials and programs around that I've come across. Okay, so I believe so.
Thank you, Professor. Please, let's move on to Kingsley. Kingsley, please unmute yourself.
Good evening, everybody. Thank you, Professor, for the wonderful presentation. I have a question based on the current projects you are doing, the plant-based research. I understand that a lot of medicinal or herbal medicinal products have actually given a good promise as a source of anti-malaria treatments, anti-malaria treatment, but malaria treatments, so to say. But I also know that there is a problem associated with standardization of dosing, and also a lot of, just like you rightly said, plants are very good source of synthesis, or they perform synthesis more than what a chemist can do in the lab. So with respect to that, a lot of inactive organic materials are also found within the plants. So how can we ensure that those inactive ones will not give a negative interaction to the active ones that are of interest? Are there any measures put in place to avoid such problems from occurring?
Okay, thank you very much, Mr. Kingsley. That's a very beautiful question. You see, I already mentioned the fact that there might be problem with standardization, okay? And that's because plants and their phytochemicals, they are too much subject to variability. You have variability geographically, if I collect the same plants in Anambra and I collect it in Osun State, they're likely going to be different when it comes to the constraints in them. Okay. If I collect the same plant in April and I collect it in June, or I collect it in December, they may also be different. If I collect it, you understand what I'm talking about. Plants, it's almost like every single plant has its own phytochemical fingerprint. Now, I've been thinking about this before we even started this project, but I believe it is doable. It is doable. There's a way we can standardize it. Let me digress a little bit. I work in the pharmacy, and I can tell you there are quite a number of herbal products that's on the shelf that are doing amazingly when it comes to giving therapeutic effect for different disease areas. One of them is Adizwa. I don't know how many of us know Daflon. Daflon is a product that we use to treat hemorrhoid, what they call PI.
Right.
Yes, Daflon has been in existence for decades. The question is, how are they able to ensure the quality of that product still? The same thing Adizwa. Adizwa is from oligospermia, men that have low sperm count. It works like magic. You understand? I'm not just saying it because the product is fine, but because I have patients that are on it and they are doing well. It's also from plants. How are those producers, the manufacturers, able to standardize? It's probably because they have a process that they're not discussing with anymore. Okay? They have a process to do that they're not discussing with the academic community. And that's because for the larger part, the academic community in Nigeria, we don't solve problems. We just publish. We publish, get promoted, and move on, we retire. That's all. We don't really solve problem. So we've been talking about this standardization for a long time, but it is doable. Now, let me tell you us about a particular product, a particular work we did on COVID-19. We got some grants led by Professor Babalola UI at that time during COVID-19 to work on some of the products that we already have in markets, herbal products, herbal health drink, and then we tried to study this other product, moringa seed. And moringa seed gave us the highest clearance of that particular virus. It was almost difficult to believe. We did the study in Germany. I was in Germany at the time during COVID-19. Now, what I want us to realize is that moringa seed is actually hepatotoxic. Okay? Even though it cleared the virus, it had hepatotoxicity. And how did we do it? The person in charge of the project, he figured out how to do it and remove that possibility, that hepatotoxic principles from it, so that the product you will have eventually will not have that toxicity. So if you come back to what we are producing, we already have an idea how to go about it. Now, for instance, let's assume I'm interested in plant AM, okay? And that plant AM has 10 different compounds, or let me say 5 compounds in it that I'm interested in, that we have shown to have very good binding affinity with the with the targets that we're interested in. But now that plant has other compounds, several other compounds that we may not need. The fact that we don't need them doesn't mean they are useless. We should also be very careful, okay? It's also possible that we have better compounds in that plant that we are not seeing yet, that they have not even discovered. But we are only interested in those 5 compounds. What we can do is that we can look at the physicochemical attributes of those compounds and work out an extraction system customized for them. I don't know if you get what I'm talking about. We work out an extraction system that can selectively enrich our medium with those compounds while reducing the concentration of the other ones. Now, it means by the time we're done with the study, we're not just going to collect plants, mix them together, and package. No, we have to refine them by using customized extractive procedures. So I think that should solve the problem to a large extent. And we can selectively enrich our product with the kind of phytochemical we're interested in. Thank you.
All right. Thank you so much for the explicit answer.
You're welcome.
Thank you. So we move ahead so fast. That guy, please, I think it's your turn. Unmute yourself and ask your question.
All right. Good evening, sir.
Hello, Shadrach. Sorry, that guy, Farm Shadrach. Because of the time, because we've taken Prof for a long time now, so that we'll not be inconveniencing so much. And so we can just take 3 questions for those who are, so no one has to be taken after the 3 people that have raised their hand now. Thank you, Farm Shadrach. Thank you. So that guy, please can go ahead, please.
Yes, sir. Good evening, sir. Good evening, everyone. Thank you, sir, for the presentation. I'll go straight to my question. And it's very short and simple, but maybe a little bit deep. The point is, I would like to ask, how do we deal with the friction between orthodox or like clinical practice and their reception to herbals? Because, okay, yes, we can, put this drug could be doing so well and put it out there in the market. But I mean, in this field, we know that a lot of doctors, they have their own somehow negative reservations towards herbals. So how do we actually deal with that reception? I think it would actually be maybe kind of an obstruction or a hindrance to the growth of this, polypharmacology towards the aspect of using herbals to treat diseases. That's my question, sir.
Thank you very much. Well, I don't believe, personally, I don't believe doctors have reservations with respect to herbals. It's not just doctors, even some pharmacists, some health professionals, okay? But what they have reservations for is the fact that the practitioners of herbal medicine in Nigeria, they are largely uneducated. I believe you know what I'm talking about. If you are going maybe from Ibadan or Lagos to anywhere, you probably will see somebody that will come and start selling a product to you. Okay, even the packaging alone will scare you, because when you look at the packaging, you can already assume the kind of care or otherwise they must have taken in producing that thing. The next question is that are they even sure that it gives any effect that they are expected to have? So that is what doctors, or let me say mostly educated people, have problem with. You understand? Now, the way people have gone ahead with herbal products, I don't want to start mentioning some names of product, but I will mention some. Let me talk about bitters. Let me talk about ginger and the likes. If you follow the production, the development of this product, they were not developed to be herbal medicine. Okay, they were developed more to be supplements, and there's a difference between the two. When you develop something to be supplements, it means you are not even showing any data of efficacy. You are simply telling NAFDAC that this product is safe. It will not kill people. And by the time you look at the information that NAFDAC will put there, NAFDAC will say that, okay, this product has been listed. It's not a drug. It's not a herbal drug. Okay, it's simply a supplement. But there's a lot of, let me say, there's a lot of fraud that is going on, whereby a company will get a listing certificate from NAFDAC. Listing means it is safe, but it doesn't mean it works.
Yeah.
So they will get the listing certificate from NAFDAC, but they will go to the public and start advertising it for all the drugs, for all diseases. You understand? They will bring ginger to you and say, oh, it will do this, it will do that. The person telling you that it's going to cure diabetes doesn't even know what diabetes is. You understand? The people that produce ginger, they probably know nothing about hypertension. So the certificate they get from NAFDAC is that this particular product is safe, but we have never studied whether it's going to work as a drug. So it is not a herbal drug to start with. I hope you understand that. Now, the kind of product that we are interested in is something that by the time you are done with with every single phase of the research project, we will have to go on to do preclinical trials and clinical trials. And usually, clinical trials are conducted by medical doctors. Currently, we have a product that we are working on the application for its clinical trials in Nigeria. It will be done in the 6 geopolitical zones in Nigeria. I'm still working on the application today, and the clinical research aspect of this product is being led by a medical doctor. In fact, most of the clinical team, I'm part of it, most of them are medical doctors. So now tell me why and how a medical doctor or medical doctors are going to have a problem with a product that they have examined in patients and they have seen to work. Do you understand my question?
Yes, sir.
They are not likely going to have problem with it because it was developed by them. In fact, what we call drug development talks about examining, investigating that particular product in the hospital, in clinics. It's done by doctors. So the moment you are able to take your products through these stages, these proper drug development stages, no doctor will have problem with it because you have data to show. Adizua is like that. There are many of the products like that. In fact, Adizua is prescribed by doctors entirely.
Okay.
You understand now? So what you see around, they are not herbal drugs. They are just almost fraudulent products. Even the people producing them don't even understand much about disease systems. So we should not have that. We should not think that's that problem that we have. If your product, the herbal product, solves a problem, no doctor will have problem with it. Because if that doctor even has that disease and your herbal product developed properly can solve that problem, he will buy it. He will pay millions for it. Thank you very much.
Thank you, sir.
Let's be fast. Onyebuchi or four, I think it's your turn. You can unmute yourself.
Hello, good evening, everyone. Profs, good evening. Professors in the house, I'm greeting.
Good evening.
Prof, thank you so much for this time, and it has been very educating and very interesting. Okay, I want to go straight to the question. My question is that first, I want to support the research work you are doing about the malaria treatment or the malaria drug discovery in process in already in pipeline. First is why I must say that I like the idea of the multi-target system or multi-target procedure or aspect that's trying to combat this malaria from multi-target. Why I believe so is that it's better off because if this product should be developed and quality maintained, it will go a long way because there's something I used to ask myself at first. I don't know whether you mentioned it earlier because I joined a bit late, but then most times I just asked myself, what could be the major reason for resistance? And for me, I think the major reason is exposure. And because I believe if a parasite is in the body or maybe any organism, let's say, if it's in the body, it doesn't get to mutate until it has been exposed to a particular agent or a therapeutic agent. And if it's not exposed to that, it doesn't know what this agent, the molecule is like, and therefore it might not develop that resistance. So, and I'm feeling like if this product is developed very well and the quality maintained, and on the other hand, it's solely protected from adulteration, definitely it would go a long way from not getting issues or not having issues and maybe mutation, because if a drug comes in, does what it's supposed to do, kill to the end, then definitely there will be no chance for mutation. Okay, or the chances for mutation will be greatly reduced. Then that's supporting your view. Then back to my question. My question is that most of the time, I just discovered that in Africa, we almost think, or in fact, we are just like the term malaria disease seems to be a problem with Africa and some Asian countries. But then this malaria we're talking about is caused, we have known the vector, mosquito is there. So why not? I haven't been coming across any effort to eradicate mosquito more. Instead, it's always more about how to, cure what the damage that it has caused. So I'm thinking, are there not maybe possible things to do? Maybe looking at ways to eradicate mosquito entirely in Africa, like the way where Europe and these developed countries are mosquito free. I know we might be talking about their climate, but uniquely for our own country, aren't there things that we might do? So if you can trade light to that, I will be very grateful. Thank you.
Thank you. Thank you very much. Well, I also believe that the mutation is usually a response to the selective pressure that chemicals like drugs exert on the particular parasite, especially when you use suboptimal dose of that particular chemical. If you're supposed to use 80 milligram and you decide to use just 30 milligram. So it's going to kill some of the parasites, but the ones that don't die, they become gradually resistant to that particular chemical. So thank you very much for that. Yes, the vector control. I said probably before you joined, a lot of things have been done in that line too. Vector control. I talked about the insecticide treated nets. Talk about the public health issue of removing how do we say it now, stagnant water from the environment, cutting bushes and things like that. But if you want to be practical, that's nearly impossible to do for the entire Africa. So even if Nigeria, let's say one state or two or three, or all the states in Nigeria decide to do that, as long as the neighboring countries to us don't do the same thing at the same time, we still have the traveling, let me say, of the mosquito back to us. So it may not be so practicable right now. You also mentioned the fact that the weather, our weather support makes them thrive. The African weather, the humid, warm weather in Africa makes them thrive well. But there have been research efforts, even with products available. The only problem is that many of us are not even aware of such products. Like you have some products, some cream, repellent cream. I believe these are products of research by different Africans, okay? Some cream that you can rub on your body that will prevent the mosquito from biting you, and then you have insecticide treated net, like we have said. And I'm also currently a part of another project. And our interest, our objective is now to control the vector, the mosquito. It's purely research. Okay, we're trying to study the life of the mosquito to see exactly how we can get them eradicated. We may not be able to eradicate them on a continental level for Africa, but at least if you have a means of eradicating them the moment they step into your house, I think it's a big win for us already. Okay, even though you may not be able to remove them completely, but if you have a product that when you put in your house, one way or the other, that is not toxic to humans, kills all the mosquito, I think it's a big win. So there are still people thinking in that direction also. We cannot just pick one area and say, okay, we are neglecting every other area. So that will be the final, I believe that will be the end to malaria by the time we are able to control mosquito. But the control of mosquito and the eradication of them is not just a life science research thing. It's more of a civil engineering thing also. Okay, most of the European countries, when you go there, you hardly see stagnant water everywhere. It's not medical doctors or scientists that did that. It's usually civil engineers that make sure that that doesn't happen with them. So thank you very much.
Thank you. So our last question. Honor, I think it's your turn. Go on.
Hi, good morning. Good evening, everyone.
Yeah, go on.
Hello, good evening, everyone. My name
Yeah, good
is
evening.
Ibrahim. Professor Olubiyi, thank you for your intensive lecture. The first thing I would like to add some things to some questions some people have asked. Someone asked about how can we sustain all these plants so that maybe they won't wear out over time. But the question I would have asked the person is that for the past years, we have been eating vegetables. We still have vegetables available. So wearing out shouldn't be how will I call it, the concern now. The concern is us getting a product that will work better than what we have before and will solve all these unavoidable questions that has been raised concerning malaria. The other one I would like to add is polypharmacology. Yeah, polypharmacology is now partly available on virtually too. We call it network pharmacology. So for some people that wants to do polypharmacology, it's something you can also go into if you want to do polypharmacology before you go into how will I call it, the lab. It make it more easier. And for people that wants to go into malaria research, there is a website where you can dock up to 43 targets of malaria at once with your compound, up to 10 compound at once. It will dock them against 43 targets. And the name of the site is called plasmodocking. So for people that wants to do that. Then concern someone asked about how do we try to eradicate compounds that are kind of toxic, but they are still in the current of the plant. There's something we call the extraction protocols. Whereas you look for protocols, for example, if you observe that your active compounds in the plant are flavonoids or terpenes or saponins, there is a way that you can only your solvent will extract only the type of compound that you need, which will involve the type of solvent you will use to fractionate your extraction. So that's how people usually do away with removing some unnecessary toxic compound from plant extract. So my my own how will I call it? My own question is that, Prof, there was a time you talk about negative you talk about molecular dynamic simulation of 100 nanoseconds. I don't know if I get you right, sir. And there was a work I did recently. I did 300 nanoseconds. This is out of malaria parasite work. I'm curious because you mentioned 100 nanoseconds, and trying to publish the work. One of the reviewers asked me to increase the simulation to 600 because 300 is too short to be analyzed. So if 300 is too short and you mentioned 100 nanoseconds, I'm kind of a little bit confused. Is the reviewer the one taking the work too serious, or I don't get it? So I'm just kind of curious because he said it's either I do about 600 nanoseconds or I should make sure I repeat that 300 nanoseconds twice. So that was his response to the paper. So I want some little clarification on that, sir. Thank you, sir.
I didn't know whether I should call you Mr. Ibrahim or Dr. Ibrahim.
Thank you, sir.
It's Dr. Ibrahim.
Congratulations on finishing your PhD in South Africa. I know that voice.
Thank you, sir.
That name. Yeah. It's good to hear from you again.
Thank you, sir.
Yeah, let me just talk about the MD simulation problem. Now, we have some reviewers who are difficult. Okay. Some reviewers, sometimes they even ask you to do things they cannot even do in their own research group. I remember when I was doing my PhD work, I was going to publish something there. That was, I think, around 2010, 2011, read about. I'm thereabouts. I did simulation of 1 microsecond. That's not even 300. That's 1,000 nanosecond. And the reviewer told me I should extend it. I should do the simulation longer. So I had to increase each of it. I'm not talking about one trajectory. I'm not talking about one system. I'm talking about almost 4, 5 different amylose systems. So I had to increase them to 1.5 microsecond. That's 1,500 nanosecond, right? Imagine doing that in 2011. Now, I was able to convince them that that was enough because I also did chemical shift calculation for the critical atoms in my protein system. So I got the chemical shift data from a professor in US, and then I now did the calculation of the chemical shift I did in GROMACS. I created a script that was able to track my trajectory to be able to check at what point that trajectory was able to converge. That's what they call structural convergence or conformational convergence, okay? At that time, it means your system has reached equilibrium. Generally, if your protein system has not reached what we call equilibrium, you cannot stop simulation. Now, it also depends on the kind of problem you're trying to solve. If you're trying to solve a protein folding problem, then you may have to do it long. Because by the time you do the MD simulation and you do maybe RMSD computation or radius of gyration or any of those descriptors, and it doesn't show a plateauing, a flattening of your profile, or maybe your profile is still rising. Maybe the RMSD value is still gently going up, or it's coming down, or it's just disturbed. Generally, it doesn't plateau. It will tell the reviewer that your system has not equilibrated. And the next thing they will tell you is that you should try to simulate longer because they want to see whether that trajectory is going to converge. You understand? So
Yes,
many of us do simulation and we don't even pay attention to the equilibration of the system. So when I see those journals, I say, okay, this has not equilibrated. You have to simulate further. But if you don't have the time to simulate further, you can also create multiple trajectories. The only problem is that you have to vary the random seed so that you make sure that you are not sampling exactly the same thing. Sometimes you may have to anneal it. It means you increase the temperature at the starting condition and then get different population of conformation to now begin new MD simulation. But in my case, I used 100 nanoseconds and that was enough for me. And I will tell you why it was enough. I was not interested really in the conformational dynamics of the complex. I just wanted to use MD simulation to decide on which particular compound would detach from binding post.
From binding
Okay.
sites. From
And
binding
the
posts.
ones would remain. And by the time I did the simulation for about 20 nanoseconds, I saw some of them detached. Okay. They literally moved away. Some of them detached and reorganized, rearranged themselves and reband. You understand? And you could see that.
Yeah.
So by the time I carried that to 100 nanoseconds, I'm not talking about just a single trajectory. I did maybe more than 300 trajectories. You understand? It's not just
Yes.
one. I'm talking about almost 300 different trajectories for 100 nanoseconds. So it's massively ensemble docking. So nobody even commented on that. They knew we did well. So that may be the reason why the man is asking you to extend. There must maybe your system has not equilibrated. So thank you very
The
much.
system has equilibrated, but I will just increase it and see what happens, sir. Thank you.
And another thing is that you don't have to always do everything reviewers ask you to do. Okay. You can debate it. You can debate them and tell them that, okay, we don't have the resources and we believe what we're trying to find in the work has been solved, has been found within 300 nanoseconds. Okay.
Okay, sir. Okay, sir. Thank you very much, sir.
You're welcome.
Yeah, thank
you,
you.
Dr.
Thank
Wande.
you.
Thank you, Dr. Ibrahim.
Thank you. Thank you, everyone. Thank you, Professor Jide. Thank you so much for the time. I think I'm handing over back now to Dr. Wande. Thank you, everyone.
Yeah, thank you so much, Shadrach, for moderating this session. And I want to say a big thank you to Professor Olujide Olubiyi, an excellent professor of repute. Thank you for coming to share your experience with us, and I believe we've been able to learn a lot. And I think this is one of this is what we should be exposed to oftentimes because we really need it for many emerging scientists that are coming up in the area of drug discovery. So we appreciate Professor Olubiyi for giving us this time out. Thank you so much, sir. And I believe so much that some of that time where we want to call you over again, because I believe many people enjoy your presentation, sir. So you'll be available to oblige.
No problem. Anytime, sir. Thank you very much.
All right. Thank you so much, bro. Okay. So yeah, I think thank you to everyone for coming. We appreciate you, and we want to wish us a very wonderful night rest. Thank you.
Thank you. Bye.
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 Thank you.
Thank you.